In this project, our group examined how caffeine is metabolized and specifically how this metabolism process changes during pregnancy. It's commonly recommended that caffeine consumption should be reduced when someone is pregnant, and we were interested in why this actually is. But before we can talk about how caffeine is processed differently during pregnancy, it's important to understand how it's processed normally in a person who is not pregnant. In this first figure, we're given an overview of the metabolic pathways of caffeine in liver cells. And as with any other molecule, there are multiple pathways that your body can use to process caffeine after it's been ingested. We will be focusing on the pathway that converts caffeine to paraxanthine, which is right here, and paraxanthine to AMFU, right here, because this is the most likely metabolic pathway for caffeine in the liver. So in this first step of the metabolic process, the enzyme CYP1A2 catalyzes the conversion of caffeine to paraxanthine, which I have highlighted here. This is the primary enzyme used to metabolize caffeine in liver cells. It's responsible for about 95% of the processing. CYP1A2 is used to demethylate the caffeine molecule, which means it removes a methyl group. Caffeine has 3 total methyl groups that can be possibly removed, and the product of this pathway is paraxanthine, which has had the top methyl group removed and replaced by an amino group, which I have circled here. The 2 minor products that are formed via similar pathways are theobromine and theophylline, but we are focusing on paraxanthine since it's produced the most abundantly. This is actually the rate-limiting step of this pathway because caffeine must be demethylated before it can be processed further. So this whole process is largely dependent on how much CYP1A2 is available and how active it is in the cell. CYP1A2 activity is determined largely by the genetics of a person, and its production is regulated by the aromatic hydrocarbon receptor, which is abbreviated AHR. So after paraxanthine is created, the enzyme NAT2 converts it to AMFU, highlighted here, and subsequently AAMU metabolites. Not everything is understood about this step in the metabolism of caffeine, so it's difficult to say for sure how this reaction proceeds, but something that CYP1A2 enzyme helps again at this stage and assists NAT2 in adding an acetyl group to the aromatic amino ring, which can be seen right here in paraxanthine. Overall, this metabolic pathway for caffeine is a detoxifying process because it converts caffeine into metabolites that can be excreted from liver cells as waste.
So to kind of go into this a little further, there was a study that was done on pregnant rats, mostly because there's not a lot of studies done on actual human pregnant women, but there was a lot of correlations with this study in pregnant rats. As you can see, a study conducted on rats highlights that CYP1A2 activity and protein expression are decreased during pregnancy. And you can kind of see that in this graph here that's pulled from this study, how the Vmax is lowered in late gestation and so on. You can see that in this graph on the upper left-hand corner, the MROD is the control. So you can compare all of these data points to that, as well as in the upper right-hand corner of that graph, the circle, the highest Vmax is also that control. You also see that the Km decreased significantly from 332 to 205 nanomolars in late gestation. So there is a significant decrease, and we're going to kind of discuss further if the reasoning behind that and if we can kind of go over that. But first, I wanted to point out there's a lot of P450 isoforms. With so many, we really need to point out that and this is from the study specifically because they wanted to make it really clear that this is these very specific P450 enzymes. The activity of CYP3A4, CYP2C9, and CYP2D6 are actually increased. And it was interesting because it was increased up to 5-fold during pregnancy, which is incredible. But CYP1A2, which is the one we're specifically discussing, is decreased. So when they were tested on the rats and all these things, they saw that there was that specific decrease. And so that really suggested that CYP1A2 is connected to somehow caffeine and how you break it down. So to kind of discuss it further, it's hard to really pinpoint what's happening. Is it the activities decreased? Is the protein expression decreased? Is there just less proteins? And I really thought the study had a lot of really good things to say about it. So just kind of going from the left side of this screen, I mean, the greatest effect was observed in CYP1A2 activity, followed by a smaller change in the protein expression, and then the least effect in the mRNA. So that's a pretty good indicator that mostly it's just the activity has been downregulated overall. This result of interest is because with most other P450 enzymes, the largest magnitude of change is observed in the mRNA, which is kind of the ones we discussed prior, those 3 prior that actually increased. So those are mostly changed in the mRNA. However, there's a huge difference between the CYP1A2 and those 3 other enzymes we discussed in the prior slide. So this is kind of saying, the study's also kind of saying like they believe it's due to post-translational regulation rather than the effects of the CYP1A2 mRNA. So kind of tricky, but they're pretty confident that it's mostly the activity is downregulated. And then despite the significant decrease in the CYP1A2 protein expression, no change in mRNA was observed in mid-pregnancy. So it's so little, this mRNA change. It takes all the way, there's a little bit in late pregnancy, but it's so minimal that it's really not, we can't draw a strong conclusion that it is due to the mRNA. So I really like this ending based on these results. It's likely that multiple mechanisms contribute to decreased CYP1A2 activity during pregnancy, which really highlights the complexities of these enzymes. But I think the strongest conclusion we can make is that mostly the activity is just decreased.
To go a little deeper into the metabolism of caffeine being slower in pregnant women, we know this is because the main enzyme that metabolizes caffeine, the CYP1A2 enzyme, is reduced in pregnancy. As you can see from the chart, at the very beginning of pregnancy, the enzyme is fully present as usual. By the middle of pregnancy, this enzyme is reduced by about 35%. Toward the end of pregnancy, the enzyme is reduced by up to 52%. The other enzyme that has been mentioned, the NAT2 enzyme, is reduced early in pregnancy and stays low throughout the pregnancy. To fully understand the effect these enzymes have on pregnancy, we can look at the half-life of caffeine. In an average individual, the half-life of caffeine ranges from 2 to 4.5 hours. By the 17th week of pregnancy, the half-life of caffeine is as long as 10 hours. It is hypothesized that the reduction in these enzymes is due to the higher levels of estrogen and progesterone in pregnancy. The placenta doesn't have the CYP1A2 enzyme at all, so it doesn't metabolize any caffeine, and a baby doesn't have very much of this enzyme either until they're about 8 months old. The half-life of caffeine in a newborn is about 50 to 103 hours. That's 2 to 4 days. A fetus has to use renal elimination to clear caffeine, which isn't nearly as efficient as metabolizing it with the CYP1A2 enzyme. A fetus excretes 80 to 90% of caffeine in their urine compared to about 2% in an adult. The large amount of unmetabolized caffeine in a fetus and newborn's body can block essential processes and cause other problems. Here are our references. Thanks for watching.